Thank you very much. And congratulations to the University of Lund on having a 350-year anniversary and on hosting this really outstanding occasion. I've greatly enjoyed this morning. So what I'm going to talk about is related to some research I'm doing on the topic that I mostly research on, which is this condition here that's in the middle, this developmental language disorder, which is, interestingly enough, a condition that is rather a Cinderella subject. I would think most of you have heard of dyslexia. Most of you have heard of autism spectrum disorder. Being in Lund, you might have heard of developmental language disorder, because Lund is a great place for research on this topic. But most of the time, people are rather unaware that there is this condition which is sitting really between the two, because it involves more than just reading, where dyslexia is predominantly just reading problems, writing problems, but not so much with the emphasis on oral language. In developmental language disorder, children have rather selective problems with starting to learn to talk, but it affects both speaking and often also comprehension. Whereas with autism spectrum disorder, there's a broader range of problems that go on to affect social interactions and various aspects of behavior. So these have typically been regarded as separate conditions, and they raise the question of, yes, the brain is amazing, but why do we get these children who have certain kinds of developmental difficulties? What is happening in their brains? And when we sort of first looked at this, the idea that was very widely around in the very early days was that it was all the fault of the parents. There were things called refrigerator parents that caused autism, was the view of some people, or that parents who didn't talk to their children. But what has become increasingly clear is that these are actually conditions that have some genetic component. And how do we know that? Well, you can find evidence for genetic influences on people's behavior if you look at family resemblance. So if you see that people who are related to each other are more similar in terms of having, for instance, a reading problem than people who are unrelated, that starts suggesting it might be genetic. But of course, that could also be a consequence of them having some kind of similar family environment. So what's very nice is the natural experiment of twins, because twins who grow up together can be either identical twins, who are genetically identical, or non-identical, who are no more similar than just regular brothers and sisters. And if you find the identical twins are more similar to each other, for example, having dyslexia or autism or language disorder, than the non-identical twins, that starts to really suggest a genetic influence. Because they're growing up together, they're exposed to the same environmental influences, so why should one group otherwise be more similar than the other? And in all three of these conditions that affect neurodevelopment, there's clear evidence of genetic influence, in that genetically identical twins are more similar than non-identical twins. And I've done some research on this, particularly in relation to language disorder, but there's now quite a large body of studies showing this repeatedly. So when this first happened, a number of us who were in the area thought that the next step would be very easy. We'd pass the problem on to the people who did molecular genetics, the people who stare down their microscopes or who analyze DNA sequences in people, and that what they would probably find is a gene for dyslexia, a gene for developmental language disorder, and a gene for autism. Alas, I have to be careful with this slide because people sometimes go away thinking this is what I'm saying. Notice the word up the top there, wrong. This is totally wrong because as time has gone on and people have tried to do this, it's become clear that nothing could be further from the truth. The truth looks a bit more like this, only this is an oversimplification. But there have been numerous genes that have been found to have some influence on language, dyslexia, autism, and I've added here ADHD, attention deficit hyperactivity disorder. There are also some environmental influences, but the relationships are very, very, very complicated. So instead of there being this nice one-to-one mapping that I suggested at the outset we were looking for, it's clear that there's what we would call many-to-one mappings, but in both directions. So you can have the same cause with rather different effects, and that's a theme I will return to later in the talk. But you can also have the same disorder, so you can have a condition like autism, and it's now clear that there's a number of very different factors that can cause that. What makes matters even more complicated is that you can have interactions between genes and environment, so that the impact of a gene might depend on the particular environment in which a child is developing. And you can have interactions between genes, again, another theme I'm going to come back to, so that it's really very complicated trying to sort out what is going on and what the underlying bases of these conditions is. And although from time to time one hears excited cries that we found what's called a biomarker for a condition like autism, so at one point there was a claim that we could diagnose autism from brain scans, these things have not stood the test of time. And in general, if you look at a brain scan of a child with one of these conditions, it's pretty difficult to tell whether they are typically developing or whether they have a neurodevelopmental disorder. And in fact, the variation often within these groups is as big as the variation between groups. So we have a complicated situation, and what on earth are we going to do? I mean, this was me pulling my hair out when I realized just how complicated it was, and started to think, well, what can we do to move forward? And there is one way of moving forward which many people are adopting, which is just to get huge samples where you've got such a very large number of people that you might be able to uncover even very tiny and incomplete effects. But the other that is more tractable is to focus on conditions where we know what the genetic basis is and which we know are associated with neurodevelopmental disorders, and to try to study them to track exactly what the path is through from genes to brains to behavior. And in this regard, I became very interested in a set of conditions where children are growing up with an additional chromosome, and in this case it's an additional sex chromosome. And I had been aware for many years that there was a very small research literature on these children suggesting that they did have rather high rates, particularly of language disorder. But relatively little had been done with them for reasons that may become clear. So in order to understand the type of thing I'm talking about, I'm going to give you a very brief primer in genetics, which is probably well beyond the level that most of you, well, much simpler than the level most of you already know. But essentially, humans have these paired chromosomes. The DNA is on one of these pairs of chromosomes, and we have 23 of these. And 22 of them are what are known as autosomes, so they are just the same regardless of whether you're male or female, or whether you come from your mother or your father. But in addition, we have these two sex chromosomes, X and Y. And as you see, they're rather disparate. The X chromosome is huge, and the Y chromosome is this rather insignificant little thing. If you've got an X and a Y, you're a male, typically. And if you've got two X's, you're usually a female. But we do get people who then have 3 copies of sex chromosomes. And this can happen in 3 ways. You can either have 3 X chromosomes, which is known as triple X syndrome or Trisomy X. That affects about 1 in 1,000. Or you can have 2 X's and a Y, which makes you Klinefelter syndrome. Or you can have an X and 2 Ys. And the interesting thing is that most of the people who are affected with these don't actually know that they have a trisomy. And so there's a large number of people out there who have these conditions but are unaware of them. So how do you get to have one? Well, this is where I would like to have a couple of volunteers. If somebody could put their hand up, you want to be a volunteer? Yeah, and I need a woman as well as a man. Yes, you? And in fact, I could have a third if you don't mind being a child who can be of either sex. Yeah, what about you over there then for this? Okay. So would you like to come up to the stage? This is just a very simple demonstration of how you can end up with 3 sex chromosomes. So these are yours, and these are yours. And if you could hold these up, you have got, we only need 3 at the moment. So, sorry, you can come later. If you could come along here. Basically, all we're going to do is show that if you have one of these from you and one of these from you, you can either end up initially as just regular male or female. So if you could each give one to her, And you hold those up, so we have a female child, and then you might want to shuffle them up a bit and then do it again, or pick one at random. Okay, now give her another one each. Okay, so now she's an XY, so this would be a male, and so on and so forth. Now, so you can have either sort of combination, but the way we get to a trisomy is when something goes wrong with the situation where normally you just have one from each. And this is the process of meiosis, where you've each got two chromosomes, but they actually come apart. So instead of that, they remain stuck together. So you get two from one parent and one from the other. So if we could start with giving these back, so you've got enough to choose from, and you can just shuffle them round again. But what I'd like you to do is to this time have two from your mother and one from your father. And we'll see what we can end up with. So what is she now? She is XXY. Which is, anybody remember what that is? Klinefelter syndrome, right? And she will be a male. And now try again with, this time, two from the father, two from you, and one from you. With that, so you've got an X and two Ys, good. And another one with two from you and one from you. See how it goes. And we've got XXY, another Klinefelters, and one more from you again, just to show the other option we can get. Two from you and one from you. I'm hoping we'll get all the options, but we'll see. We have. You probably did it in my favour. So we've now got a Trisomy X. Thank you. And thanks to my volunteers. Your names are? Florent. Louise. Florent, Louise, and Amanda, a round of applause for them. Thank you. So that's just showing you how we get to this situation. But what impact does it have? Because I've already told you that most of these people don't actually realize that they have this additional chromosome. So does it have any impact on you at all? Some of the earliest information that we had came from studies where they screened newborn babies. Now, this is a huge undertaking, because I've shown you that only about 1 in 1,000 people have this condition. And when they did screen the newborn babies, they did find about 1 in 1,000, but they then tried to follow them up and see how they turned out. And what they found was that most of these children were attending mainstream school. They didn't have major problems, but they had increased educational difficulties all the same, especially affecting language, in all 3 trisomies. Now, that was many years ago, and one reason why we don't have much more research since then is partly that these sorts of studies are very expensive to do, but there's also questions as to whether they're really ethical, because you can imagine if you have a new baby and then somebody says, oh, we found an extra chromosome, you're not quite sure what that means, and it can be quite unsettling. So nothing was done for many years, really. There are a few other studies of various kinds, but not a big amount of research until this lady, Patricia Jacobs, got in touch with me, suggesting I should do a study. I had had some dealings with her before. She's a very eminent geneticist who was responsible for discovering a lot of these trisomies. And she was getting concerned about a very interesting thing, that there were some studies out there, but they were always of people who had discovered in childhood or adulthood that they had an extra sex chromosome. And she was worried that this was not a very typical or unbiased sample of people, because the way you typically find out that you've got this is because you've got problems. So you're having some sort of difficulties, and somebody decides to test your chromosomes. And she said that we were missing an opportunity, because that might not last very long, because at that time in Britain, in our National Health Service, people were routinely getting screened if they wished for Down syndrome, which is a much more serious condition where you've got 3 copies of chromosome 21. But at the same time, they were detecting sometimes these sex chromosome trisomies. And so she suggested that there should be a study of this relatively unbiased sample by somebody to look at the neurodevelopmental outcomes all these years later. And so we did this study. We got funding from a couple of good sources, and we got marvelous cooperation from the clinical geneticists in the UK. And so we were able to take a look at what went on. And most of what we found wasn't very surprising. This is written up as quite a large study with a lot of detail in it. But the sort of thing that we found, this is exactly consistent with what was found in the 1960s, in that there's this increase in children who, in all three trisomies, having what are called special educational needs, which in the UK is a fairly general term, meaning you need some extra help at school. This is the rate in the national data, so you can see for boys and girls, it's higher for boys than girls. But as you can see, and these are their brothers and sisters who don't have the extra sex chromosome, so you can see these people have got a higher rate, but by no means all of them have got problems. Group 1 is the prenatal group we had focused on, but we also got a lot of people who wanted to take part in the study, but who were later identified. And it basically confirmed what we had suspected, is that if you look at those people, you will find higher rates of problems. These tend to be people who have problems and therefore find out they have this condition because they get tested. So that was confirming that it was important to make that distinction. But the surprising result was here on autism spectrum disorder. Now here, we had to rely solely on parental report. We didn't see the children for this study, so we just asked them, has your child been diagnosed with a number of conditions? But this was surprising to us because it had never been commented on in the earlier studies that there was this increased rate. It's still a minority of children, its numbers are quite small, but even in these prenatally diagnosed children, the boys about between 1 in 10 and 1 in 5 were having a diagnosis, and again with the postnatally diagnosed XXX and XYY, again we see it in the boys, whereas in the general population at the time we did this study, those were the figures that you would get. We think this is not as interesting as it sounds, because we think it's relating to different criteria that are now used to diagnose autism, which are much broader. So when the early studies were done, autism was diagnosed in a very strict way and really only used for children with very extreme difficulties. So we think this is probably not as exciting as it might seem to discover the link, but it did excite me because it suggested that maybe things that give you a risk for language disorder are not that different from things that give you a risk for autism. Whereas in the general literature and in clinical practice, people had always stressed that these were really rather different conditions, and it seemed rather that there might be differences on a continuum rather than as separate as all that. So we've gone back and over a great many years taken time to recruit further cases, again using a similar design, but this time we wanted to actually see the children and do some hands-on assessment, and I won't have time to say much about exactly what we did here, but this is just to give you the broad idea of the nature of the study. So we recruited some children who were prenatally diagnosed and some who were diagnosed later, and again we've got all three trisomies here represented, and we were looking at their language skills, communication, laterality is something I'm particularly interested in, and neurodevelopment. And again, what we found was really challenging the idea that these neurodevelopmental disorders are really very separate, because this is just a very rough and ready way of representing things categorically, and we've got a lot of very continuous measures that we could present here, but this is just to show you how much overlap there is between these categories that are typically regarded as part and parcel of the same thing. So as you can see, these are the children who've got only autism in blue, but then we've got some who also have evidence of language disorder or dyslexia. A lot of them have language disorder, which may or may not co-occur with these other two conditions, and so on through all of these three trisomies. You're seeing children who are really intermediate between our traditional categories or have some signs of all of these. And this is true regardless of which chromosome is the extra one. So this is interesting in its own right for sort of suggesting the boundaries between these conditions may be less precise than we thought. But it also makes you start thinking, well, what is the mechanism that means that something about having this extra chromosome is affecting brain development to put you at risk for neurodevelopmental disorders, regardless of whether it's an extra X or an extra Y? And this is actually very helpful in helping you home in on where the difficulties might lie, because there's rather few candidates for things that would fit the bill, really, for being a plausible cause. Having an extra bit of DNA, it doesn't have to be a whole chromosome, is generally not a good thing in terms of genetics. Most of us have got great chunks of DNA that nobody else has got duplicated or deleted and so on. There's a lot of peculiarities of DNA out there, but often they are not really doing very much because they're not involving important genes. But if you have duplications of genes, it's just as bad sometimes as having something missing, because you get the wrong level of protein being produced by these genes, and that really can mess up the development of the brain. So the fact that trisomies are not good is understood, but why are there such similarities? And there's an interesting thing about the sex chromosomes, which is really very, very special and makes this even more interesting. And that is that although I've shown you females having 2 great big X chromosomes and males having a big X chromosome and a little Y chromosome, you might expect that there'd be a lot more differences between males and females because of that difference in the amount of DNA and the number of genes that would be expressed. Well, nature has found a way to stop that being an issue by a process known as X inactivation. So most of the second X chromosome in females doesn't function. It's inactivated, it curls up into a sort of ball, it undergoes a process known as methylation. And therefore, in effect, females typically have 1 active X chromosome, except for these bits at the end, which are known as the pseudo-autosomal region, where everything is expressed, and so you've got, they behave just like the autosomes in that regard. You can actually see this in the body of a cat, this X inactivation process. There's a lot of fascinating research on coat color in animals, because coat color is often influenced by genes on the X chromosome. And what you're seeing in this cat, which is a calico cat, is patches of black, patches of orange, patches of black, patches of orange. These are patches of skin that have developed out of an original cell in the developing embryo that has either had the black version or the ginger version of coat color inactivated. And then the other cells grow from that, and it spreads. So you're actually seeing, you know this is a female cat because it's a calico cat that's got this coat color pattern. So that's X inactivation. But it makes you realize that if you've got 3 X chromosomes, this explains why the impact is really much less than you would see in something like Down syndrome, where you've got 3 full copies of chromosome 21. Here you've got most of the extra 2 chromosomes inactivated, but you've got these bits on the tips that are in 3 versions, have got 3 rather than 2 copies. Exactly the same for the boy with Klinefelter syndrome will have these extra bits. So it must be these extra bits that contain something that, when overexpressed, isn't so good. Now, the Y chromosome, as I've said, has not got much on it, mainly genes for determining sex, but it has got pseudo-autosomal regions as well, and there's no inactivation, but it's not really needed because there's not much going on the Y chromosome. Well, that's my way of putting it, probably not too accurate. Anyway, so what we're doing here in saying it that if we're looking for something that might account for these problems in both the girls with XXX, the boys with XXY, and the boys with XYY, we want to find some gene that is located on this pseudo-autosomal region, probably, because that's where the overexpression is happening, where there's what we call homologous forms, the same gene in effect on both the X and the Y chromosome. It's expressed in the brain, because that's where things are happening. And ideally, one where we've seen an association with a neurodevelopmental disorder. And Pat Jacobs, who went on the hunt for this, came up with a wonderful candidate called neuroligin 4. Now, neuroligin 4 meets all those criteria. There have been studies showing that mutations of this gene are sometimes associated with autism and similar conditions. The literature's a bit up and down on this, but that's true of most genetic association studies. But it did seem to be a plausible candidate that having an extra dose of this gene might play a part in the story of why we are finding the similar problems in all 3. But we're not just finding similar problems in all 3. We're finding something very strange if you approach this from the point of view of, we're going to find something wrong with these children, because there's massive variation within each of these trisomies in what you see. You may have, if you were really awake, noticed that there were some children who had no diagnosis of any kind when I did those Venn diagrams. So there are some children who appear to be totally unaffected. It's a minority, but they're there. Others had much milder problems. This is just illustrating it with some data on a continuous scale known as the social responsiveness scale, which is quite a good measure of neurodevelopmental problems in general. And if you're right up the top, the odds are you've got autism. And this is the typical range. And being high is bad. But these are individual data points for the 3 trisomies for the prenatally diagnosed, postnatal. And as you can see, once again, these guys are really much more extremely impaired, and we think that's just an artifact of how they're selected. But what you can see within all groups is that in all groups, there's a problem, regardless of which trisomy you have, but there's huge variation right through from children who seem to be doing just fine, don't have any difficulties, from those who have quite severe problems. And they've all got the same type of chromosome difference. So how do we get such variation? And that's what we're currently trying to find out. So I don't have an answer to it, but I have a speculation which we're following up now, doing some DNA analyses. But I think it's useful to tell you about my speculations. They're usually wrong, so I'm likely to be found wrong, but just because it's a rather different way of thinking about the causes of these disorders, which I think we're going to need to embrace, moving away from the idea of we're looking for a gene for this or a gene for that or a brain region for this or that. So I put up a speculation warning because I don't want you going away thinking I've shown this to be true. But the idea is that it may very well be that what you're seeing here is this additional gene product that you're getting from this neuroligin, or maybe from something else, but we think it's that, may just amplify the effect of other variants elsewhere on the genes, not on the sex chromosomes, but other genes elsewhere on the autosomes that normally have very small effects. Because most of the other things that we're finding that are part of normal variation, you can find things that have effects on development, but they're typically tiny. But what about if they were amplified by having this neuroligin present? Why should they be amplified? Well, there's one reason why that might be the case, and that is that if they are in the same basic network, they're working together to do things like support brain development in a particular circuit. This is a diagram that I couldn't begin to explain to you, although I'm sure there's people in the room that could. But it's just showing you here, my old friend, the neuroligins, interacting here in the synaptic cleft with neurexins. And what you're then seeing is 2 different genes, both of which are important for synaptic development, and one of them is a neuroligin. The other happens to be a gene that's been shown to be associated with language disorder, but which is perfectly common in the general population. And the effect it has, the link with language disorder, is really tiny. It just sort of nudges your language skills up or down a little bit in the general population. But the idea is this might have a bigger effect if seen outside the general population. And I want to close just by illustrating how this might work, and I need 4 more volunteers. So, anybody want to take part? Yes, you? It doesn't matter what, you? Can we have a couple of girls? Couple of girls? Yes, you, please. Another one? No more girls? Yes. If you'd like to come up to the stage, yes? Right. So, what's your name? Axel. Hello, Axel. Axel, you're going to have the great advantage of being the person who has neither this genetic risk factor for language disorder nor a trisomy. So all you have to do is to, as fast as you can, pick up marshmallows, 1 at a time. You can use both hands if you wish, but only 1 at a time, and put them in there. And we're going to see how many you can get in there. Okay. Your name? Ali. Ali, you're going to be the person who's actually got the normal variant that makes you not quite so good at doing these things. So you're going to do exactly the same task, but with 1 hand behind your back. Okay. And you are? Amanda. We're going to ask you to do exactly the same thing, but you have got the misfortune to have an extra dose of neuroligin, and that corresponds to wearing a rubber glove. So apart from that, you can use both hands if you wish to put them in, but this hand is handicapped. And you know, your name is? Kaisa. You've drawn the short straw because you're going to have to wear a rubber glove and put the other hand behind your back when you're doing this. And you're going to have to put as many of those in there as you possibly can when I say go. So are we all ready? Yes. Ready, steady, go. I have to say I've never done this before. And stop. Actually, I think Kaisa did rather better than I thought she would. Which just goes to show that even with a rubber glove and only one hand, you can do rather well. But she did get rather less. I mean, she did about this. Yeah, there's fewer in there than that one. And as expected, you managed to get most because you were relatively unhandicapped. But that is the end of the... That is the end of that demonstration. I hope it makes it at least stick in your minds that there is this conjunction of problems that you can have, any of which on their own may not have a very serious effect on you, but if you put them together, I should have made it a harder task for you, obviously. I should have given you a great big gardening glove or something. But nevertheless, that was great, and I'd like to thank our volunteers for hoping to get that in your head. So a round of applause. And I think since you've picked these up, you should take them away and eat them, if you would like to, or share them with your friends. Thank you. You can leave the rubber gloves behind. Okay. So thank you. Right. So really, this is just to wrap up, to say what have trisomies taught us. These children are fascinating, and of course, they're fascinating in their own right. It's a common enough condition that we want to understand it. But they also are fascinating for what they reveal about how flexible the brain is and in developing, and how it's not what one of my American colleagues called a cookie cutter thing, whereby if you've got a particular genetic condition, you'll always have exactly the same thing. It's hugely variable. And the thing that it really suggests is that genes don't work in isolation, so that if you have a variant that has a small effect in one context, it may have a big effect if there's a double hit on a functional network. But thank you very much. That's it.
Thank you very much, Professor Bishop. Now, will I get to use this again? Oops. Do we have any questions? Hands in the air. I'll start out to see if you warm up, you just put a hand up and you'll get this one. If this, having an extra X chromosome or a Y chromosome, is occasionally the cause of autism spectrum disorder.
Yes.
Do you think that if we started testing for that, would those children or grown-ups get a different type of treatment? Would they need something else? Or would this still be just a curiosity? Like, okay, interesting. So
yeah, I thought you were going to ask something else, which I'll briefly answer because it often causes confusion.
Okay, please.
This is not a common cause of autism. Right. So this is one of the difficulties that when you sort of say there's this association, people think that all children with autism may have this. Autism can arise from a great many different things, and this is just one of them. But would it lead to a different treatment? In general, I think not. And most of the quest for good new treatments for children's various neurodevelopmental disorders, people come along with all sorts of ideas. If only we can get at the underlying problem, we'll be able to treat them more effectively. But in practice, so far, my reading of the literature is that the most effective treatments are the ones that look at the presenting problem, regardless of the cause, and try and work with that. So if you've got a child who can't read, you should work on reading. You shouldn't be messing about with trying to hone their cerebellum or something. And similarly with autism, I think children with autism can already be very variable. And I think the people who know most about working with autism would argue the thing to do is to focus on what are the presenting problems with that child.
Right.
Sometimes it does just help the child, though, if it's known that there is this biological factor involved, just in terms of it becoming slightly more acceptable. Some children are happy to know that they have an explanation for being why they are.
Right. Did I see a hand? Yes, I did. Just a second, sir. And there you go. Oh, that wasn't so bad.
You frequently hear...
Hold it closer up, please. And we'll
okay,
hear
you frequently hear when scientists are working with models and it doesn't work, they always explain that the reason is that there are compensatory mechanisms. And I think this goes very well in line on what you are saying, compensatory mechanism. Mm-hmm. Could you maybe elaborate a little bit on that, because it seems to be very close to your topic?
So mechanisms that might compensate for this.
Yeah, I mean, I think what we've focused on a lot is we're looking for genetic variants that may slightly increase your risk, say, of language disorder, and where you may end up with maybe slightly misplaced neurons is one theory, or something else. But I think what we have ignored is looking for what might be called the opposite of risk factors, which would be protective factors. So that the one thing about the brain is it often has many ways you potentially can solve a problem. And I think my take on this is that there are children where, because one system is strong, if the other one is not so well developed, they have the ability to compensate. But I think it is the reason I said speculation alert is that until we have hard data, this is very hand-wavy. But we do have DNA on these children, and we're going to look at some of these genes that have already been actually linked to language disorder. And the prediction would be we should see a much bigger effect relating to that gene on things like their language outcomes. But whether it's two sides of a coin, whether it's that some children have something that actually actively impedes learning, or the others have a different way of going about things, I think are two sides of the same coin.
Thank you.
Thank you. Yes, we have a question over there, rather far at the back. So throw us, yes, if you look and then you throw as far as you can. Absolutely. Let it have a...
Yeah.
There you go.
Okay, can you hear me? So I was wondering if you could comment on this idea that autism is a form of extreme male brain, and that is why you have more autists in the XYY group.
I think it's the extreme male brain theory is a very elegant theory in the sense that it can neatly explain a lot of phenomena, but at the same time, it's been subjected to a lot of tests that have not supported it. So it has been linked to high levels of testosterone. And one of the odd things about the boys with XYY is that they don't have particularly high levels of testosterone, as far as we know, when people have done. I haven't studied that. So I think it would seem to fit, but the problem I feel at the moment with the theory is that my guess is that if somebody were to sit down and do a very objective meta-analysis of all the studies looking at testosterone in relation to autism, the evidence wouldn't look as strong as sometimes is suggested. Diplomatic way of putting it.
Thank you.
Thank you. More questions? I was thinking of how you said that it probably wouldn't make a whole lot of practical difference if you started testing, for example, children with autism for an extra chromosome. But having said that, could you say anything about how far do you think we are from treatments that actually try to sort of block the effect of having, like this, an extra chromosome or extra genes? Is that
I think we're a very long way, but I think that what we need to do is to really understand now at the molecular level how this is operating. But I think everything I'm saying is suggesting it's going to be everything's more difficult, but it's not going to be just a case of finding, oh, you've ramped up one thing and therefore it has this effect on the developing brain. I think it is, because it's going to depend in different children on the genetic background against which these effects occur, it's going to be really quite difficult to get there. But I think that is the solution. I mean, I showed at the beginning that, you know, we're trying to go from genes to brains to behavior. And we really need to work on all of those parts of the puzzle. So we have got very little with these children at the moment at the brain level. There's a little bit of research, but it's rather old. But it would be quite interesting to, if we could peer in the brain at a much more detailed level than we've been able to do so far, and see what impact the trisomy has had on how the brain is functioning, not just how it physically develops.
Right. One more thing, just would you just briefly develop on what happens when people with an extra X chromosome have children of their own?
Remarkably, if you're a girl, it seems to not affect your fertility. There has been a recent, very recent, in the past 3 or 4 weeks, somebody has very, very rapidly got hold of all the data from something called Biobank, which is a huge British study, which I took part in, I'm a subject. And they were able to look at the chromosomes on thousands and thousands of women who are now in their 50s, 60s, and find those who did have a trisomy, none of whom were aware of it. And they were able to show that these women were having children, and I think that they went through menopause a little early, on average, but basically the general view is that they have children, and the children do not have the trisomy. The boys with an extra X are typically infertile, and they don't go through puberty, and they are much more likely to be picked up because of that or because of infertility in adulthood. So there it does have an effect. And again, the XYYs, we know very little about them, but what we know suggests that they are normally fertile and that their children don't have the extra Y.
Interesting.
It is interesting. I mean, it's not very well understood how and why.
We might know more about this in the future, but
Yeah.
for now, thank you very
Okay.
much, Professor Dorothy V Bishop.